Hello everyone, welcome to Deepti's biology class. So we are starting with the process oogenesis. So what is oogenesis? Oogenesis is the development of egg or ovum from ovary. So this is the structure of ovary and we will be seeing how every month ovum is released, or else in every 28 days the completely developed egg is released, right? So this development in this ovary we will be studying in the form of flow chart first, then we will come back to that diagram, right? So there is a speciality in oogenesis when compared with spermatogenesis. Spermatogenesis starts at puberty age or puberty level, but oogenesis starts when a female embryo is in the womb of the mother. That means in the fetal stage of female embryo, it starts developing the egg, right? Starts developing egg. In between it stops, again it restarts at the time of puberty. So let's see how it happens. Now, so here in these are different names which are given. You have to be very careful when you name it. So oogonia is a diploid structure which is having 46 number of chromosomes undergo number of mitotic differentiation. When it happens at the fetal life of the female embryo. So whatever new cells which are produced, it is termed as primary oocytes. Primary oocytes. Primary oocytes are also diploid cell, like C chromosome number which is given there. So what happened till now? Oogonia, or else we can say that this is the mother cell which is going to be developed in future as the ovum, right? Undergo mitotic division. It is equational division. As a result, number of oocytes, primary oocytes are produced. Now, this primary oocyte, these all are diploid in number. Let us trace one primary oocyte and we will see what happens to that, right? One primary oocyte, what will happen? Now, after this primary oocyte formation, this is already we know diploid in structure, right? Now, there is first meiotic division which starts, but at prophase 1 it get arrested. So understand, first meiotic division starts, but it get arrested at prophase 1. So prophase 1 we know it is the beginning of the division, right? When it is arrested in the sense is that no further development. So just primary oocyte started meiotic division, but it is not completed. So this fetus is going to be delivered. At the time of birth, this dotted lines indicate what it indicate? It indicate arrest, right? It is arrested. So this line indicate now the meiosis is arrested, no more development. So during childhood there is nothing. So what is actually there in the ovary right now? There are primary oocytes which just started division, meiotic division, but got arrested in prophase 1. Now, at the time of puberty, this arrested stage resumes. So what is the meaning of resume? Resume means restart. So now what happens? So when we say that at the time of puberty, what happens? A girl child get menstruation. Just prior to this puberty, what happens? Just before, or else we can say at the time of puberty, this meiosis 1 continues, and this primary oocyte, the same one, right? This primary oocyte 1 we have taken as example, get and this completes meiosis. When it completes meiosis, what happens? Two cells are formed. Which are they? So we can say that meiosis 1 got completed. Now meiosis 1. Meiosis has two steps, right? Meiosis 1 and meiosis 2. So meiosis 1 got completed. What it forms, we call it as secondary oocyte. So there is a difference here. As we know, this development of the embryo takes place, or the new baby is taking place in case of every organism or almost all organism inside the female body. Now suppose if there is a cell which is diploid in nature and undergo meiosis. if it divide equally, and as we know, the human uterus is made in such a way that it can hold one baby, that is proper, right? So if all these cytoplasm is going to be equally divided, this one is going to be wasted later. Why? Because only one ovum or one egg is going to develop. To prevent that, as we know, ovum get fertilized with the sperm within the female body, and the body is made in such a way that one egg fuses with one sperm, right? So what happens is that to store that entire cytoplasm and all, in case of female during oogenesis, maximum cytoplasm is stored in one cell, and there will be a small cell which is formed with very limited cytoplasm. This will be destroyed or reabsorbed by the body itself or ovary itself, and this is the one which is going to develop further. That is the reason you can see there is a polar body which is very small and a secondary oocyte. So what happened here? The primary oocyte started meiotic division when it was in the fetal life. Now it got arrested at prophase 1. The baby is delivered. During childhood there is no development, right? Birth time there is no change. Always remember, there is no new primary oocyte or primary whatever cells are there is formed after birth. In a female, all these primary oocytes which are formed till the time of birth. Once the baby is born, there is no new primary oocyte is developed. All primary oocyte is ready at the fetal life itself, and they undergo meiosis 1, that means first meiotic division, and arrested at prophase 1. Now at the time of puberty, what happens? This first phase of meiosis completes, and the cell divided or the primary oocyte divide into two, which are the first polar body and the secondary oocyte. Now the secondary oocyte is haploid in nature. Now you understand, because it is meiotic division, the two daughter cell will be having half number of chromosome. Now remember why this division is unequal. As we know, one cell is going to be developed into or developing into future ovum. So the maximum cytoplasm, nutrients, everything is stored in one. If it is equally divided, since the body cannot develop both, this will go waste, right? To prevent that, all these nutrients or all these cytoplasmic content is stored in the large cell, we call it as secondary oocyte, which is a haploid cell. Is it clear till now? Now, very important thing you have to remember here. Every time whenever there is menstruation, secondary oocyte is the one which got released, not the ovum. We normally say that when there is ovulation or when there is just before the menstruation, what happens? Ovum get released. If you look at this diagram, even here it is written ovum. But actually what it is releasing here, whatever it is released, we call it as secondary oocyte. I will tell you why do we call it as secondary oocyte. This is for every class students to avoid confusions. We use the term ovum, but we cannot call this structure as ovum. So if the question is asked, see, this process we call it as ovulation, releasing of the egg cell or the female sex cell from the ovary, we call it as ovulation. You can see this cell get released, right? During ovulation, which cell is released? Is it ovum? No, it is not ovum. What it is released is secondary oocyte. So secondary oocyte gets released. This is what is going to be fertilized with the sperm cells. As we said now, that from the ovary it will be collected by the fimbriae, then passing through the fallopian tube, it wait in ampulla, it get fertilized with the sperm. Do you remember when we have studied female reproductive structure, we said where the fertilization and all takes place? That is actually this secondary oocyte. Now when the secondary oocyte will become ovum? Now what is going to happen here is that this secondary oocyte is released at the time of ovulation. It will wait for a sperm to get it fertilized. If not so, it goes out as menstruation. Now, just I am making one ovary, right? There is the fimbriae and this is the fallopian tube. And here will be the uterus. This is a part of female reproductive system, just imagine. So, from this ovary, what it releases now? it released this secondary oocyte. Now the secondary oocyte will wait in the fallopian tube. Now if it get a chance to fertilize with a sperm, now the second meiotic division resumes. That means meiotic division, meiosis 2 will resume only if the secondary oocyte get fertilized with or on encounter of sperm. So understand, that means now this, once the sperm comes closer to this secondary oocyte, or suppose if this is a secondary oocyte, right, and this is the sperm, the moment the sperm encounter this ovum, second division takes place. So we will get a second polar body and ovum, which is also termed as the ooted, which means every time whenever there is ovulation, what is released? During ovulation, what is released? We call it as secondary oocyte, which is a haploid cell. It will be released from the ovary. It will wait in the ampulla region. If it get a chance to fertilize, or when it encounter a sperm, it continue division and it completes meiosis 2. That means meiosis as meiosis 1. At that time, see, this was the meiosis 1, first meiotic division. Now second meiotic division can only takes place if the secondary oocyte encounter the presence of sperm. The moment sperm touches it, it will be undergoing. That stimulation is going to make this the second division. The ovum is formed. If there is no sperm there to encounter, or there is no copulation, no sexual reproduction, what happens? Ovulation takes place. What it is released is secondary oocyte, not the ovum. So understand the difference between the stage at which secondary oocyte or ovum can be formed. Normal situation only up to there. See, if a girl child is there, till now there is no copulation, or else we can say that till now, just we can hypothetically take that this child is not having any sexual intercourse. So what happens? Every time there will be the secondary oocyte which is released. Now the secondary oocyte in the female reproductive system, it when menstruation happens, it is removed. Now the secondary oocyte, once it is released, if it encounter a sperm, what happens? That result in meiosis 2. So there will be further division. The ovum, which is the completely developed ovum, will be there. Now fertilization will take place. Secondary oocyte is not undergoing the fertilization. Instead, immediately it will be converted into a fully developed ovum. That ovum is going to fertilize with the sperm. So here you have to remember, it is encounter. That means the moment the sperm come in contact with the secondary oocyte, it undergo meiosis 2 and it become ovum. Now the sperm will get fertilized. So please get this difference. Secondary oocyte encounters with sperm. Now what happened? The secondary oocyte which encounters with the sperm, this secondary oocyte will be converted into ovum. How? By undergoing meiosis 2. Now this ovum immediately get fertilized with the sperm and it will be converted into zygote. So this complete thing, many facts are there. Now you have to understand one thing. When it starts at fetal life. All primary oocytes are produced at fetal stage, right? Later what happens? It is arrested. At the time of birth, no more new oocytes, new cells are not formed. Many get destroyed from the ovary. Childhood time there is nothing, but puberty time it resumes, and the entire reproductive life, what happens? Already ready primary oocyte which got arrested at prophase 1 keep converting into secondary oocyte. If sperm is there, it will be immediately become ovum, then it fertilizes with the sperm. So you have to understand, in your age what happens at the time of ovulation, what is actually released? It is the secondary oocyte, not the ovum. But you will not find a diagram where it is written secondary oocyte and all. Why? because for junior kids and all it will be lot of confusion if it is written secondary oocyte here. Instead they write it as ovum, the ovum which get released. But remember, if the question is asked at what stage egg is released from the ovary during ovulation, it is the secondary oocyte which get released, right? So just see the factors now. Oogenesis is the process of formation of mature female gamete, right? It is initiated during embryonic development stage, and millions of gamete mother cells or oogonia, right? Oogonia are formed in the fetal ovary. No more oogonia are formed or added after the birth, right? This first step which we have written here, this step will never happen after the birth. All these things happen before the birth. Oogonia form primary oocytes by number of division. Now this oogonia, or else we can say that the primary oocyte, right? We know oogonia undergo mitosis to form primary oocyte. Now this primary oocyte start the process of meiosis and get arrested at the stage of prophase 1. Now these prophase 1, those cells which get arrested at the prophase 1, we can call it as primary oocytes, or else we can say in this way also, oogonia which undergo mitotic division, there will be primary oocytes which are formed. This way you can say, or else you can say this is the oogonia which keep dividing, it become more number of oogonia, but the thing is same because the division is mitotic division. Now what happens, each oogonia or primary oocyte is undergoing which type of division? Meiosis 1, but it get arrested. So either you can call it as primary oocyte, right, primary oocyte, or else you can even say that, see, are you getting that point? Here I told you, see, the cell keep dividing. Now I told you oogonia which undergo mitotic division, you can call these new cells are either oogonia, or else you can say that these are the primary oocyte. But remember, even at primary oocyte, what happens? First meiotic division will take place and it get arrested at the phase, which phase? That is the prophase 1 phase, right? Now each primary oocyte gets surrounded by a layer of granulosa cell. This is very important. Now the cell which is, just imagine this is the primary oocyte, a cell with a nucleus, right? A cell with nucleus. Now some cells are there which keep adding on this structure. Then only development will take place. As it comes, I will tell you the different names will be there. Now surrounded to this primary oocyte, what happens? A layer of cell get added. These cells are called follicles. What is the name of this cell? These cells are termed as follicular cell or the follicles. Since it is around the primary oocyte or primary oocyte, we call it as primary follicle. So what is primary oocyte and primary follicle? When around primary oocyte, a layer of granulosa or follicular cell get surrounded, we call it as primary follicle. These group of cells we call it as granulosa cells or follicular cells. Now what is the speciality of these cells? These cells are nothing but they are capable to produce the female sex hormones called estrogen, that we will come in a minute. Now a large number of primary follicles get degenerated between birth and puberty. So these are the cells. Now you can see here, these were the primary oocyte. Now around the primary oocyte, what happens? You can see these round structure, right? This follicles which get attached. So we call it as primary follicle. As it keep growing, it will become secondary follicle. Then again it is developing, it is going to become tertiary follicle. As this way it keeps increasing. Now at puberty, there are only about 60,000 to 80,000 primary follicles in the ovary at the time of puberty, which means there are millions of oogonia which were undergoing development, but at the time of puberty almost all of them get destroyed and there will be only 60,000 to 80,000 primary follicles in the ovary, which means only they will be there to develop. Again, many will get destroyed or many will remain as it is without any development. You can understand, in a woman or in a girl we can say puberty starts around 12 to 13 years old and it will last up to 52 years. So in these many years, every year there will be 12 month or 12 times menstruation can happen maximum, right? So you understand, every time only one is required. So in a year there will be 12 egg need to be released out of this. So you can calculate how much is going in waste, right? Now these remaining primary follicles get surrounded by more layer of granulosa cells as well as new theca. They form secondary follicle. Now what happens? As I told you here, what happens? There was an egg cell, surrounded by this egg cell there were this follicular cells, right? Or else we say that granulosa cell. Now surrounded to this granulosa cell, a new layer get developed. So when there was granulosa cell which was added in the beginning, we called it as primary follicle. Now surrounded to this entire structure, a new layer develops, we call it as theca. So we call it as secondary follicle. So if you look at this diagram, you have to understand here, right? So there was oogonium or whatever was there, primary oocyte was there. Then when granulosa cells or follicular cells surrounded it, we call it as primary follicle. Now around primary follicle, what happens? A new layer of theca got added, like more layers of cells are added, then we call it as secondary follicle, right? Different stage we are talking about. Now, so as it is written, there will be new theca which is added. Now these become secondary follicle. Now secondary follicle then give rise to the tertiary follicle. Now these tertiary follicles are having a speciality, we call it as anthem. Now when you look at this tertiary follicle, you can see some fluid filled cavity here, right? This yellow colored one. This yellow colored fluid we call it as anthem. This is the fluid filled cavity. So actually in this structure, where is our egg cell? This slightly reddish cell, can you see? This was in the center here. If you look at this diagram, it was in the center. See this red color thing which you are seeing, this much structure is actually the egg cell. See, it is keep dividing, it is towards one side now, right? And this is the anthem. Anthem is present, as that is a fluid filled cavity. Later what happens? This egg cell which will get released, right? So this is the development stage. So there was primary oocyte, right? Then primary oocyte, when it formed granulosa cell around it, we called it as primary follicle. Then when there was theca around it, we called it as secondary. Then when there was fluid filled cavity called anthem, we called it as tertiary follicle. So understand the difference. So primary, secondary, tertiary. In primary there was granulosa cell. In secondary, granulosa plus theca. In tertiary, granulosa is there, theca is there, and there is a new thing, we call it as the anthem, fluid filled cavity called anthem, right? Theca is organized into two layers. Now as it develops, what happens? That layer of theca, see, just let me make it into little more deep, right? So there was this primary oocyte. Around the primary oocyte, when there was these layers of cells were there, we called it as primary follicle. Around primary follicle, what happened? There was another layer, we called it as theca. Now what happens? So we call it as tertiary. Now this theca will develop into two layers. So one dotted and one complete line, right? So there will be outer and inner theca layer. So that is what written here. The outermost layer was theca, which is divided into two. That is inner theca, and or that means we can say inner theca interna and theca externa. The primary oocyte grows in size and completes its first meiotic division. So you have to understand, during this time what happens? There will be first meiotic division. there will be first meiotic division. So these completion of first meiotic division, that means these all things are happening at the puberty time. At the puberty time, as I told you, primary oocyte got arrested at the time of, when it happens at the time of birth, at the phase of prophase 1. Now at the time of puberty, what happens? This primary oocyte undergo becoming primary follicle, then secondary follicle, tertiary follicle. All these things are happening at the time of puberty, right? And it completes the first meiotic division. It is an unequal division forming large secondary oocyte and a tiny first polar body, that we have seen in this case, right? When the first meiotic division completes, there will be first polar body and secondary oocyte, right? Now this, which means once it comes up to here, there is the secondary oocyte which is formed. Now the secondary oocyte retains much of the nutrient-rich cytoplasm because it was an unequal division. Now the tertiary follicle changes into the mature Graafian follicle. So see, what is the difference between oocyte and follicle when we talk about? Oocyte surrounds the follicle, right? See, if it is the primary oocyte, when there was a layer of follicle which surrounds here, this type of layer which surrounds here, we called it as primary follicle. Now when it is, what is the difference between secondary oocyte and secondary follicle? So when, suppose if this is the secondary oocyte, surrounded to secondary oocyte, what will be there? These layers of follicular cells are there, and the fully developed oocyte cells, we call it as Graafian follicle. So what is Graafian follicle? Whenever there is meiotic 1 division completes, what happens? There will be the secondary oocyte which is formed this way. Whenever there is an unequal division, there is a large secondary oocyte which is formed and a tiny polar body. Now the secondary oocyte is going to retain almost all the nutrient-rich cytoplasm. Now the secondary oocytes are present in the tertiary follicle, right? These are formed as now the tertiary follicle. Now see, development is taking place. This is actually stage 1, stage 2, this way you can write. As the development takes place, when the tertiary follicle is there, all these structure is going to have the secondary oocyte. Tertiary will be there. As the secondary oocyte surrounded by the layers matures, we call it as Graafian follicles. Now what is the significance of this Graafian follicle? This Graafian follicle is completely developed secondary oocyte it contains. So what is there in this Graafian follicle? Graafian follicle meaning is that ripened follicle. In this ripened follicle, what will be there? The fully developed secondary oocyte is there. Now not only that, this particular secondary oocyte develop a new layer around it, we call it as zona pellucida. That we will see what is this zona pellucida. So what all things will be there? There will be, see, if this is the secondary oocyte, first there will be this zona pellucida will be there, then there will be all these granulosa layer, theca layer will be there, all these multiple layers. Once we will separately study the structure of secondary oocyte or the completely developed ovum, there it will be understanding what is zona pellucida. Now the Graafian follicle now ruptures to release the secondary oocyte, in bracket it is written ovum. Now you know what is the difference between ovum and secondary oocyte, right? Secondary oocyte can be called only as ovum if it encounters with the sperm. Now the Graafian follicle, this one, completely developed one, ruptures to release the secondary oocyte from the ovary by the process of ovulation. So this releasing of the secondary oocyte is termed as ovulation. So when the process of oogenesis is asked or development of ovum is asked, you no need to write all these stories and everything. You have to write, you can draw this diagram plus this flow chart you can use when the question of oogenesis is asked. Even if you are explaining, you can use this flow chart to explain the process of oogenesis, right? So you must have a trace on this chromosome number. Here you have to write which phase it get arrested, the prophase 1, and if there is sperm to encounter, then only meiosis 2 will be there. Hope this is clear. In the next lesson we will see what is the structure of, or what is the further development of the ovum. Thank you so much.